Today we're going to be discussing taste and smell, both the reception of taste and the perception of taste, and then the reception of smell, and then the perception of smell. In general, tastants are the chemicals that are tasted when you're tasting something, and odorants are the structures that you're tasting, that you are smelling when you are going through olfaction, when you are smelling something. So remember that both taste and smell work through chemoreceptors, and the actual chemicals we're referring to are tastants and odorants. Now, both of these things happen in aqueous environments. You're going to have tastants that interact with neurons. You're going to have odorants that interact with neurons. But these can't happen in a dry environment. So tastants must be dissolved in saliva, and odorants must be dissolved in mucus. So this is just a really good example of something that you should probably already know, is that most physiological, or all physiological functions only happen in aqueous environments or water-filled environments. So you cannot sense taste or you cannot sense smell unless you have an aqueous environment for those things to happen in. So tastants are just tiny little molecular particles of the food that you're eating, and odorants are just tiny little molecular microscopic particles of the thing that you're smelling. Alright, so let's talk about taste. What we're looking at here is a tongue, of course, and what we refer to as taste buds is probably not what we actually think they are. And when you stick out your tongue, you see all these dots on the tongue. These are not actually taste buds. They're called papillae. Remember, a papillus is a nipple-like projection. So there are 3 different types of papillae on your tongue. These ones out here, there's some in the back here, and there are some along the side here. We're not going to spend a lot of time on the different types of papillae, except to say that the taste buds exist along the sides of the papillae. So, for example, right here we're looking at a velate papillus, and what you'll notice is that along the sides of it you'll have like a dozen of them along the side, and they'll probably be radiating along different edges of this structure. So along the sides of the papillus you'll actually have a whole bunch of taste buds. So here we see all these little blue dots here. These are taste buds, and if you look at it blown up, it looks like this, and it actually looks like a flower bud that's looking to flower. So that's how these got their names of taste buds. All right. So when you look at the taste bud, it's along the side here, and remember that tastants are going to be dissolved in saliva, and what's going to happen is that those dissolved structures, those dissolved chemicals, are going to make their way along the side of the papilla, and they're going to interact with the taste buds. And the way they do that is the taste buds are actually buried within epithelial cells, but there is one small hole here called a taste pore. So some of the tastants with some of the saliva are going to make their way down along the edge of the papilla and make their way into the taste pore. So each taste bud consists of, well, actually 3 different types of epithelial cells. There are stratified squamous epithelium that make up kind of the protective walls around this entire thing, this protective coating. There are also gustatory epithelial cells. Look at all these things in here. All these long purple-looking things in here are gustatory epithelial cells, living cells. You can see the nucleus here, that are actually the sensors of taste. Additionally, there are basal epithelial cells. Basal epithelial cells. Now, these are basically the stem cells of these structures. They are cells that are ready to differentiate into taste bud gustatory epithelial cells whenever they're actually needed. So you don't actually get to the neural structure until you see this one right here. So here we have a cranial nerve. And here are the dendrites sticking out of the end of the cranial nerve that are ready to send that information back to the brain. And so these little yellow structures here, they're part of the nervous system. But what actually senses the food, again, are the gustatory hairs. Are the gustatory epithelial cells. And the part of the gustatory epithelial cell that actually senses these are these structures here called gustatory hairs. So the gustatory hairs are going to project from the tips of the gustatory epithelial cells, and you'll see that they kind of are, looking towards or sticking out towards that taste pore, because this is the only place where they're actually going to be able to sense any kind of tastant coming into the mouth. Okay, so these are vulnerable cells. They're epithelial cells, which is nice because epithelial cells in general are always regenerative. These ones particularly so. These can actually regenerate every 7 to 10 days because taste buds are subject to friction and burn, and they will need to be regenerating. Pardon me. They will need to regenerate on a pretty regular basis. Okay, so that's the job of the basal epithelial cells, to just be there to grow up when these other guys, these gustatory epithelial cells, die off because of friction or burn. Okay, let me see where we're at. Hush, kitty. Shh, shh, shh. Okay, so we've covered the basal epithelial cells, the gustatory epithelial cells, the gustatory hairs. Those are the kind of the terms you should be paying attention to there. All right. Now, the tastant that comes through here are actually going to cause these cells to depolarize. When they come through here and they interact with the correct cell that they themselves are able to depolarize, then these guys depolarize, and then the signal is sent back to these dendrites, and then it's sent back to the brain. So we'll talk about that, what happens next in a moment. But let's talk in a little more detail about these taste bud cells. So it turns out that there are some taste bud cells that are, these gustatory epithelial cells that are sensitive to particular things. So we have what we call bitter gustatory epithelial cells, salty, sweet, umami, and sour gustatory epithelial cells. And those particular ones will only be sensitive to the things that actually cause that particular sensation. So let's talk about those. If you have, there are 5 basic taste sensations. The first one is sweet. Sugars, saccharin, alcohol, and even some amino acids will depolarize the sweet gustatory epithelial cells. So they will interact with these little gustatory hair cells, and they'll cause depolarization of this gustatory hair cell, which will then allow the signal to be sent back. Right? Salt, metal ions. So this cell here is what we call a salt gustatory epithelial cell. It is going to be sensitive to metal ions such as sodium ions and chloride ions and potassium ions. Sour. These green guys here are particularly sensitive to sour, and this is going to be sensing acids and especially hydrogen ions in particular. Bitter, the bitter ones will be sensing alkaloids such as quinine and nicotine. And umami is the one that a lot of people haven't yet heard of. It's only really been discovered, I don't know, these particular cells have only been discovered in the last 15 years or so. And the sensation of umami really didn't come about until about 110, 120 years ago. Or I should say the manufactured version of the umami taste. Umami is a savory, meaty flavor. And this is elicited by the amino acid glutamate. So if you hear of monosodium glutamate, the infamous MSG, this causes particular cells, these umami-sensitive cells, to depolarize. Okay, generally speaking, it is thought that every taste bud has all of these cells represented within it. Although it seems as though different localities on the tongue are particularly sensitive to particular tastes. So it could be that the makeup of the taste buds themselves may have a higher proportion, for example, of sweet gustatory hair cell, gustatory epithelial cells, or they may have a higher proportion of the bitter sensitive ones. But it seems as though all taste buds have representatives of all 5 of these tastes within them. All right, let's talk about the perception of taste. So there are two cranial nerves that actually send the information back to the brain. Cranial nerve 7, which is the facial nerve, cranial nerve 9, which is the glossopharyngeal nerve. These carry the impulses directly from the taste buds in the tongue to the solitary nucleus of the medulla. So here we have your medulla oblongata, and that is the first place where information is carried back to. And that makes sense if you know where the position of the facial nerve and the glossopharyngeal nerve are. In addition, the vagus nerve seems to carry some information from deeper in the throat. This is not specifically a taste-sensitive structure, but it may have some chemosensitive properties where it may be sensing danger, or if you just tweak the vagus nerve, it actually causes an emetic response or a regurgitative response. So vagus nerve is cranial nerve 10. So all three of these nerves carry the information to the solitary nucleus of the medulla, and then it is carried to the thalamus, where a lot of information is sorted out, actually not just about taste, but about a lot of things. And from there it is sent to the gustatory cortex to be tasted, to the hypothalamus, and to the limbic system. Remember, we talked about having an emotional response to your food, amongst all kinds of different other parts of the brain. So your taste is going to be sensed at all is going to be perceived at all of these different places. So that is the perception of taste. All right, let's move on to smell then, olfaction. Again, we're going to be talking about dissolving odorants. If you smell bread baking, what's actually happening is tiny little microscopic particles of that bread are making their way through the air, dissolving themselves in your mucus right at the top of your nose. Let me show you a picture that shows the mucus itself. Here is your mucus. So here we have little bits of bread that are dissolving themselves in the mucus, making their way to the olfactory cilia, part of the olfactory sensory neurons, and then they will be sent, up through the axons to cranial nerve 1, the olfactory nerve, the olfactory bulb, and then sent back to the brain. Anyway, so the organ of smell is going to be the olfactory epithelium, which is this entire structure in here, which covers the superior nasal concha. So here we have the nasal conchae. Here are these are bones or parts of bones that are set up to kind of swirl air around so that it makes its way upward to the roof of the nasal cavity, because these olfactory neurons are only in the roof of the nose. Many smells actually pass right through, into the throat, but some of them will make their way up. It's a very sensitive organ. So those of them that do, and if you sniff, you kind of are, you know that you'll be able to smell things better if you sniff them. And the reason you can smell things better is because you're kind of forcing the air upward to the roof of your nose. Okay, so this is the organ of smell, is the olfactory epithelium. The olfactory receptor cells are really unusual. If you look at these guys, here's one right here. Here's its dendrites, its body, or its soma, and then it makes its way. So it's a bipolar neuron, if you know what that is. And it makes its way, it sends its axon up here to interact and eventually send the signal to a mitral cell. Well, olfactory receptor cells, there are going to be millions of them in your nose. And if you take a nice scanning electron micrograph, such as this one, of the olfactory epithelium, what you'll see are tons of cilia. These are called radiating cilia. So this is one of the unusual characteristics of these neurons. These radiating cilia are very weird. These are cilia that don't really move very much. They just kind of sit there in the mucus and wait for odorants to come to them. So as far as cilia go, most cilia don't work this way. Most cilia in your body, or in any animal's body, will work by moving something around or helping the animal to move or something like that. So these radiating olfactory cilia are really unusual. Olfactory neurons themselves are regenerative. They can regenerate about every 30 to 60 days. There's only 2 or 3 other neurons that are regenerative on a regular basis, but these guys are extremely regenerative. They can regenerate every 30 to 60 days, and the reason for that is that they are the only neurons that are actually exposed to the outside of the body. So they are buried in mucus here, but you see those radiating olfactory cilia are actually quite vulnerable in their position. They're basically exposed in the nose. They are also very sensitive. They're very sensitive. You only need a few odorants to actually cause an olfactory neuron to depolarize. So when we take a look at, well, first of all, these are odorants. That we're kind of showing that this particular flower, most smells are very complex. They'll have dozens or hundreds of different odorants within them that the body kind of mixes together and identifies as one particular odor. But the odorants themselves tend to be very separate and very discrete. So here we're showing just an example of 3 different types of neurons that are sending out projections. We have red ones and purple ones and green ones. And what you'll notice about these is that at the level of these olfactory cilia, and at the level of their, of all of their dendrite complex, all of their protein receptors, the things that actually sense odorants, they're all the same. So this purple guy here only has one type of protein receptor that senses odors in all of its dendrites and along the entire length of its dendrites. So it's only going to be sensitive to the one odorant. And I guess that's probably why there are millions of different neurons in there, is that they have to be sensitive to many different types of smells. Could be you'll have a neuron that is sensitive to a particular odorant that you never actually encounter, or you don't encounter over the life of that cell. And it'll never actually be used. It'll never actually be depolarized. But it's there in case the odorant comes along. Okay. So another point that I want to make about olfactory receptor cells is that they are very, very sensitive. It only takes a few odorants to interact with these particular receptor proteins in order for the cell to depolarize. Now, the cell is going to depolarize, and this is going to be on your test. This picture, I'm going to show you a blank of this and ask you to label it just like it shows up in the practice exam with a little bit of extra information. With a little bit of extra question. There's going to be a bonus question on there. What we're looking at here is a G protein coupled receptor, a metabotropic receptor. What we've studied in the past, they're called ionotropic receptors because there's a receptor that receives, we've always talked about neurotransmitters. And that neurotransmitter binding to the receptor is going to cause the opening of a channel that is buried right within the receptor itself. Now, take a look at the difference here. It kind of works the same way because when you look at the start of this, what you'll have is an odorant binding to a receptor. You notice there's no channel here, it's just a solid protein buried in the membrane. Look all the way over here. A whole series of things is going to happen that's eventually going to open a channel. And at the end of the day, that's the whole point of the thing. There's a receptor there's odorants that bind over here, and there is an ion channel that opens over here. So this is called a metabotropic receptor system, or a G protein coupled receptor system. Because look at this. Here we have our receptor, and here is a G protein. So it's coupled or tied to this G protein. As soon as the receptor receives a couple of odorants, it's going to cause this G protein to move down the membrane to interact with this enzyme here called adenylate cyclase. Adenylate cyclase is kind of a general structure that causes things to happen, especially this, which is the conversion of ATP to cyclic AMP. Cyclic AMP then goes on to interact with this channel and cause it to open from the inside. Kind of the lock and key situation from the inside that then causes sodium and calcium to flow into the cell. So I'm going through this very quickly. I would like you to take a closer look, which is why I'm asking you about it on the exam. In general terms, metabotropic receptors or G-coupled protein receptors are usually in place because they amplify a signal. It's one of the reasons why these olfactory neurons can be so sensitive. You can have just a few odorants causing a depolarization of the cell because the signal is amplified. So that is usually the case with G-coupled protein. It's kind of like a cascade of things happening within the cell. It doesn't necessarily have to happen in the membrane itself. It could be happening, like it could be mediating a number of structures to be phosphorylated, for example, within the cell and so on. Anyway, so the point that I'm making is that you don't necessarily need a channel to be attached to make something happen within the cell. Sometimes the G-coupled protein receptors can actually be very, very powerful, much more powerful than a standard, ionotropic receptor attached to a channel sort of a thing that we've studied in the past. All right. So as far as the the way that smell is perceived. Here we have, so all that stuff I was talking about a moment ago is happening at the level of the olfactory cilia, all those depolarizations and so on. So say a cell is depolarized, it's going to send the signal onto the mitral cell, and from the mitral cell then the signal is going to be sent back to the brain. This is the olfactory neuron, cranial nerve 1. And some would say that it's not a typical, or it's not actually a cranial nerve at all because it never actually leaves the brain. You'll notice here, this is the cribriform plate of the ethmoid bone. Remember this guy? With the olfactory foramina that allow those axons from those epithelial cells to move up into the cranial nerve here. What happens is everything that is outside of the central nervous system is down here. Once you get up through these olfactory foramina, this is actually kind of the brain. This is actually the brain. This is the frontal lobe up here. This is the prefrontal cortex up here. This is the frontal lobe. So cranial nerve 1 is right under that. It's basically attached to it. So these are not typical cranial nerves at all. Some would argue they're not actually cranial nerves. Okay, so the mitral cells will then send impulses through the cranial nerve 1, or the olfactory nerve, to the olfactory cortex, and from the olfactory cortex, additionally, signals are sent to the hypothalamus, to the amygdala. Remember, the amygdala is responsible for emotional memory, and also to the limbic system. The limbic system is responsible for the feelings of emotional involvement. But the amygdala is very interesting because this is the part of the brain, I believe, that is responsible for the fact that when you smell something, it will make you remember something. The sense of smell has been renowned to be very closely tied to memory, and especially in emotional memory. So the fact that smells are sent directly to the amygdala probably has a lot to do with that. So if you remember something, if you get a scent of something obscure, you're walking through a park one day and you smell a flower that you haven't smelled in 20 years, and all of a sudden you're brought back to that garden that you walked through when you were 10 years old with your parents or something like that. This can be that strong. You can actually put yourself right back in those situations. So smell is very closely tied to memory because of the amygdala. Oh my gosh, I almost forgot. This is what I was going to say about this. I do apologize. I'm sorry I'm out of order here. When the sodium and calcium come into the cell, the sodium causes depolarization of the cell. The calcium causes the cell to become habituated. Remember that remember that olfactory neurons habituate easily and quickly, that you can become very insensitive to a smell very quickly. And that's the job of calcium. So when calcium flows into the cell, its job is to adapt the cell to the smell, and the job of sodium is to depolarize the cell.